Okay. So we move on now to the second flagship. So Rupert Luck, head of IT at EMBL. Over to you.
Thank you very much for introducing. Thank you, Domenico, for that impressive talk. Let's see what EMBL has to demonstrate and then to present. So, as you heard, I'm Rupert Luck. I'm head of IT services based in Heidelberg for EMBL. I'm presenting the setup of the flagship use case for EMBL, which is the large-scale genome analysis facility that we set up for preparing and for computing and analyzing genomic data on the cloud on demand, which is analyzing on a big data process in the cloud. First of all, EMBL, for the ones who haven't heard of the institute, EMBL is a European molecular biology laboratory, which is based, which is an intergovernmental research organization similar to CERN. We are supported by 20 member states, plus one associated, Australia. We are one of the world's most foremost life science institutions. We are pretty international, 1,500 staff and more than 70 nationalities. We are based with the headquarters in Heidelberg. We have other outstations in Hamburg, Germany, Grenoble, France, close to Rome in Monterotondo, and many of you might know the European Bioinformatics Institute in Hinton, which is hosting many of the life science databases. EMBL's mission is, one of the missions is to carry out basic research in molecular biology, and in that respect, under the headline of information biology, the scientists study how we come from single molecules to organisms, and how do we understand, for instance, the development of organisms or the complexity of organisms or phenomena such as aging or disease. In that respect, we see that information biology produces a lot of data. There's a lot of data generated by technologies such as imaging, microscopy, computational biology, next-generation sequencing. I'm talking about that more, or modeling. There's terabytes, tens of terabytes coming to our disk every week, and they, of course, require a lot of high HPC compute, and we want to make sure that this is efficiently used. However, not always the project activities are very constant, so we have variations over the time. So we study cloud computing in order to make sure how can we provision in the future elastic and on-demand IT resources, in particular high-performance computing, to European scientists within or outside of the EMBL lab. With respect to the actual use case, I'm presenting the EMBL flagship project on Helix Nebula, the genomic assembly and annotation in the cloud. We are focusing on the analysis of DNA, basically results from next-generation sequencing experiments. As you all know, our DNA is the DNA is one of the key elements to understand life science and phenomena in life science, and so it is very crucial to understand what are the important elements, what are the important genes, what are the important regulatory elements on the DNA in order to understand these phenomena. So this is a very long molecule. It's a 3 billion base pair long molecule that you need to understand, and the technology that helps you understanding and to uncover is sequencing. So within the recent couple of years, there's a dramatic revolution in terms of the technologies that are used to sequencing. And today, it is very easy to analyze the whole genome of a person, the whole genome in just a couple of hours. So basically, more or less one day. The technology has become very affordable, and that has certain effects. So it is very widely used in all areas of research, being that in academia, medical research, in pharmaceutical drug development or studies, or agricultural research. And it has also helped to understand, for instance, things like the German EHEC outbreak, or it helps us and researchers to understand variation in human genomes in projects such as the 1000 Genomes Project, or in cancer research in the ICGC project, or more complex, even more complex projects where they analyze 10,000 projects, the 10K Genome Project. So the technology is affordable in a sense of in one sense, but there's a downside. So we have a part that is in the lab where we can easily prepare, where we can extract DNA from an organism, being that a human or an animal or a plant or whatever. Put that on a slide and then sequence that in the lab. But what these sequencing machines, very powerful sequence machines, what they generate, what they spit out is not the long strand. It is very, very small fragments. They are of the size of 100 base pairs, more or less. And with compute-based technologies that are called assembly and annotation, we need to further solve a puzzle, and we need to aggregate the individual pieces to form the long strand, and then in the step of annotation to see where are the regulatory elements, where are the genes, what genes are there, how are they treated, how are they spliced in order to understand the biology behind. So with the technology being very, very popular, very affordable, you see exponential data growth. That's from our lab. You see that across different labs in all different areas. And basically, the instruments, and we are by far not a very big lab in terms of sequencing. We have decent capacity that serves the lab's needs, but we already have 30 plus terabytes coming to our disk only from sequencing, and that requires quite significant compute power and storage capacities in order to deal with that data and to analyze and to make sense from the next generation sequencing. So if you look further in terms of what is the global picture of sequencing, there's close to 9 million species across the world as an estimate, and there's some 7 billion people on the world, which, if you think in terms of personalized genomics and everybody having their genomes being analyzed and to answer questions, am I susceptible to a certain disease, or is a certain disease treatment, is that relevant to my genomic background? So you need to study you could study these genomes, and I appreciate this slide is probably a little bit outdated already, but what is still true, these technologies, if you take all the sequencing capacity that we have worldwide, and we have different large centers, for instance in China, and there's hotspots, there's exabytes per year which are generated in terms of data from sequencing, which need to be analyzed, stored, shared, and things. So there's maps that you can follow. That's basically on a daily basis. They they represent individual sequencers that are popping up each other day. And as I mentioned, they are very, very powerful data generation devices. So the idea here is really to democratize the access to large-scale genome analysis facilities, and to build an on-demand large-scale genome analysis pipeline and service to facilitate whole genome assembly and genome annotation based on Helix Nebula, the Science Cloud services, and offered as a cloud service. And then maybe with the possibility also to aggregate with data and resources and applications and tool from other organizations, being that from the European Bioinformatics Institute or from other resources. The key software components that we are implementing in our software and that we have tested throughout the different phases of the Helix Nebula pilot phase during the last 2 years are basically the two bioinformatics components, which is the assembly pipeline that is based on the SGA software from Jared Simpson from the Sanger Institute, and the annotation pipeline, which is Embl, the Ensemble pipeline. And then there's more infrastructure-related components, basically, on the one hand, the shared file system, which we implement on the basis of Red Hat's cluster file system, to offer really broadband file system capacity for high-performance computing in the cloud. And then there's a component which is called Star Cluster. That is software that has been developed within the MIT, and that offers you a very on a button click programmatic approach to start clusters and to launch full clusters in the cloud. So they start the individual VMs, they aggregate the individual VMs, they bind them together to the shared file system, and they make sure, and Star Cluster makes sure that you also have a workload scheduler in the cloud. And that picture probably illustrates this. So Star Cluster on a button click helps you to launch any sort of cluster you define, or the customer basically, that's the NGS customer that sits and defines, that submits their data in the order of 100 gigabytes coming from the sequencing experiment, submits into the cloud. That is being stored on the shared file system that has previously or at some point been generated by Star Cluster eventually, and the computing infrastructure that you can tailor exactly to your demands, whatever demand in terms of timing, capacity needs you might have. I should mention, and that is a slide which I stole from Phil yesterday evening. I should mention there's one component that is absolutely essential to us, and that is really helping us to work with the Star Cluster software. That is the EC2 bridge. So within HNX, we have access to an HNX to an EC2 Amazon Web Service interface, so that we can run the Star Cluster software successfully on Helix Nebula, and make sure that we can launch individual small or large clusters on the infrastructure. I think Phil has already mentioned, and I want to, in addition, point out that this is, of course, that is a further motivation for other customers, for future customers who are probably working with other software, being that Star Cluster or other software, working in the Amazon context, and probably a motivation also to try out Helix Nebula and the HNX platform, so that they can very easily take advantage of the user interface and make sure that their software is running. I have been, in previous events, I've already been showing that from the previous tests, we have quite decent results from the Star Cluster-based computing, and we were able to show several thousands or hundreds of thousands of jobs being running on let's say 50-node clusters. We have been showing very stable, very high stability of the cluster FS file system. That is very important because we have very long-running jobs in some areas. So the infrastructure we have really allows us to auto-provision any sort of clusters. We were able to do end-to-end tests of our bioinformatics pipelines, and we used, of course, real-world genomic sequencing data that was provided by scientists, and helped them to analyze and to study their genomes. There was, I mentioned, hundreds, thousands of individual jobs, very heterogeneous, some of them very quick, very small, very parallel jobs, some of them running long for over a couple of weeks, and the cluster stability I mentioned already. Within the HNX deployment, we appreciate that the development of the EC2 bridge took a while, and it is only available to us since very recently. And so what we had done in the actual HNX deployment so far was certainly to show that we can launch EC2 clusters, Star Cluster-based cluster deployments, and we were also showing, and this is fully automatically fully accessible to the end user, so that the client can really can go to the web interface, to the web portal, start a cluster, and launch their software. And of course, what we also found, the initial performance test were well matched the pre-HNX findings being the ones that we carried out during the pilot phase. And I would, let's say, at the end, towards the end, I would like to show you a little bit, to give you a little bit of a feeling. How does it feel? How does it look like? What are the technologies that we have implemented that makes the life of the user easy in order to do the analysis? There's the Embl NGS Cloud portal that was developed constantly during the last 2 years. And of course, during the recent time, we have heavily invested in beefing that up and trying to make sure things work. And I'm sure I can probably show you a couple of videos in order to demonstrate how things work. So what we see here is one of the components, which is the file manager. So we have a built-in facility in the web interface that allows users to browse their own desktop or a desktop where the NGS data is sitting on their own, in this case, a Mac. So you take your file, hopefully your NGS data, put it into the cloud, and then the transfer automatically begins. And that's possible with very large files. That's building on the Globus toolkit, and it will allow the users very easy to transfer large data sets. This is time-lapse. It's not very fast. You see here a local DSL line facilitating the transport. But you can also, what you can do, what helps very much. I mean, I mentioned that the data sets are very large in some areas, and so you could probably see broken data transfers. This software makes sure that the data transfers are resumed, and you can do that in parallel. So it will be very easy for the users to use. The other thing I would like to show is how easy is it to start a cluster, which is basically coming from here. So we have a first look at the dashboard in the HNX dashboard, and by hitting by just going on the initialize cluster, you start a cluster, and at one point very soon, after 3 minutes, you see the first instance being up, and then you start there's additional instances started. The full integration takes place with the file system, with the establishment of the workload scheduler, which is the Sun Grid engine. And so after some additional 3 minutes, you have your, in that case, 16 total nodes of clusters being fully deployed and ready to accept annotation pipeline or assembly pipeline calls. If you follow that on the slipstream interface on the HNX, you see the 16 instances that are launched, and so that's as easy as that. And of course, it's up to you, it's up to the user to define what is the size that you want in terms of the individual VMs. Here we had 16 VMs. You can do that with hundreds. That's done without any additional effort. Then you do pipeline runs. That's a video that we took last year already to illustrate the individual phases. So that's the annotation. So basically, it helps the user to define their runs. That's also time-lapse. It takes a little bit more. In some areas that takes hours, or it can even take longer, depending on what kind of complexity you choose in terms of your genome. But this is just to illustrate how responsive the whole interface is. The technology behind that is WebSockets technology.